introduce our two very nice speakers for today, Javier Sánchez Martín, and I will try to say properly, Zygmunt Orlovskis. Sorry for my pronunciation.
Yes, that's correct.
So Naomi, the floor is yours. You have the opportunity to introduce them.
Okay, so nice. So as always, we start with a little bit of an introduction on how specifically this working group webinar works. It's nice to see that there's still quite a few that were able to join, because it came to our attention quite late as well that it's a holiday in some countries, actually also in Belgium, so I should have known. But well, yeah, anyway, so yeah, welcome indeed to our third webinar with speakers. So the entire goal of these webinars is indeed to get to know each other and to really stay connected with each other. As well as Rafa, I don't know if you're saying something or if I can hear feedback through your microphone.
Yeah, you can.
Okay. So indeed, it is we're supposed to kind of establish future collaborations as well, and kind of know everyone that is involved in the working group too of this cost action. So what is the format? So we want each lab that is participating to kind of provide a global overview of their unique research expertise, as well as the facilities that they have available. It is a bi-monthly meeting, and it's always at the end of the month as well, always on a Friday and at 12 o'clock. So this is a very important specification as well in Central European time, importantly. So every session we estimate is going to take approximately 1 hour with 2 speakers presenting each time. Every time we can expect a presentation of approximately 20 minutes with 10 questions or 10 minutes of questions as a follow-up. Talks will always be recorded with permission of the speakers, and you can join the webinar always using the link, so it will always go to the same room as well. If there's any questions or you're interested in speaking as well, you can always let Rafa or me know, because we're still looking for speakers for the second semester as well. So please let us know if you're interested. And that brings us then, of course, to the overview. So we've already had quite some nice speakers, and today we're going to listen to, and I'm also going to try my best for pronunciation right now, to Dr. Javier Sánchez Martín and to Dr. Zygmunt Orlovskis, which are going to be our speakers of today, and we're going to start with Javier. So let's start with a brief introduction. So Javier conducts his research at the University of Salamanca, Spain. His research centers on how crops, especially cereals, defend themselves against both environmental and pathogenic stresses. Over the years, he has transitioned from field-based studies on disease and drought resistance to cutting-edge molecular work on immune receptor genes in cereal pathogen interactions. And his current focus, combining genomics, transcriptomics, and translational research, lies in understanding how Trichoderma, a beneficial fungus, primes plant immune responses to enhance resistance against biotrophic pathogens. This includes exploring the role of Trichoderma-induced microRNAs as potential plant vaccines and dissecting the molecular dialogue between crops and microbes, with the ultimate goal of developing innovative strategies for climate-resilient agriculture. So without further ado, Javier, the floor is yours. When the slide comes on, Rafa gives you the power.
Thank you, Naomi, for the presentation, and thank you to the Root Benefit Cost Action to present. My name is Javier Sánchez, and I'm presenting on behalf of Enrique Monte, that cannot present, and is very sorry, but I try to do my best. Then now for the presentation, Rafa, we share through the platform. Okay, thank you. Okay, then the talk is entitled Trichoderma, a beneficial fungus that first hits and then asks. We are in place in Salamanca, and here you have the general aspect of Trichoderma with the two states, the asexual and the sexual. On the left, you have the anaform form. We are talking about a mitosporic fungus that is reproducing asexually. And you can see in the microscope with these structures, the conidiophores, that are basically branched filaments, where you can find different chains of spores. And these spores are quite important. Sorry. Okay. these spores are quite important because allows the fungus to reproduce asexually in a very fast way, and this has implications from an agricultural point of view. And in the right side, you have the sexual phase of Trichoderma that is called hypocrea, and here you can see these perithecia, these sac-like structures where you are going to find where you are going to find the ASCII. Then everything in our group started with the PhD thesis defended a few time ago by Enrique, almost 4 decades ago, where you can see how Trichoderma arsianum is, or it was, a powerful biological control agent against Phoma betae, the causing agent of the sugar beet dumping off. And basically, Trichoderma is capable to control this pathogen through 2 main mechanisms. From one side, antibiosis, and from the other side, the mycoparasitisms. Then in the case of the antibiosis, there is the production of antimicrobial compounds that inhibits the pathogen. And basically, you have to see this like kind of a chemical warfare between Trichoderma and the pathogens. And from the other side, Trichoderma is also capable to directly, physically attack pathogens through mycoparasitisms. Then the beauty of Trichoderma is that it's capable to control pathogens combining different methods like the antibiosis or the mycoparasitisms. And here, for example, in the arrows, you can see the direct contact sites where the Trichoderma arsianum is capable to physically attack and eat in somehow the pathogen. This thesis of Enrique, it went from the lab to the field. This is an aspect very relevant for our group, and here in somehow this photo marks a historic moment because this was the first biocontrol trial at industrial scale that was done in Spain, thanks to pelleting the seeds of Trichoderma in collaboration with a company where the sugar beet seeds were coated with Trichoderma and were protected against the pathogens. We know that in the current framework of the Green Deal, the European Union wants to reduce by 50% the use of chemical pesticides. It wants to also reduce the amount of the nitrogen fertilization that is imposed in our fields. From the other side, also, we want to reduce the sales of the antimicrobials for the farm animals and promote the organic farming. And Trichoderma is capable to support 3 of these 4 pillars. Then in the context of the Green Deal, Trichoderma is really a tool to, from one side, reduce the use of chemical pesticides, reduce the amount of nitrogen fertilization, and promote the organic farming. And this is what we are trying to answer in our lab with different projects that we have at national and international level. In this case, for example, we are using with Trichoderma to increase the tolerance of wheat plants in water stress conditions. This is a project led by Rosa Hermosa. From the other side, we are trying to unravel the priming of Trichoderma against pathogens attack. This is a regional project where Enrique and myself, we are the PIs. We are also involved in other international projects, more from a genomics perspective, like the one led by Irina Drusini. And some of our results are making the difference. We have isolated from one side, from my side, more specifically, some immune receptors that are directly involved in controlling resistance against biotroph pathogens, and in the case of more Enrique, Monte and Rosa, in the case of Trichoderma, how we can use this fungus for biotic and abiotic stress conditions. Then, four are the main mechanisms through which Trichoderma is able to act as a biocontrol agent. The first one is direct competition. Basically, what Trichoderma is capable to do is to colonize the plant roots, occupying the space and consuming nutrients very faster than the pathogens, and is basically a direct competition that put the Trichoderma on the lead against the pathogens, and somehow is protecting the plants against the pathogens. We have mentioned before that the mycoparasitins, the direct fight against the pathogens, is another tool that Trichoderma is using to control the pathogens. Also, the antibiosis, where, well, through the production and delivery or secretion of different antimicrobial compounds, is capable to suppress the growth of other fungi. And something that is quite impressive is that Trichoderma is also capable to induce systemic defense. That is allowing the plants of tomato, for example, to stay healthy against the attacks of Botrytis cinerea, one of the most common diseases affecting tomato. Our lab is pioneer in the molecular identification of, for example, the genes and the corresponding enzymes encoded by Trichoderma that are involved in mycoparasitins. And here I just put in an example of the different genes and enzymes that were identified in the lab against Botrytis cinerea, and you can see the effect controlling the pathogen by Trichoderma arceanum is really fast. In just 7 hours, is capable to wipe out the pathogen using different hydrolytic enzymes like glucanases, catenases, and proteases that are directly attacking the cell wall of the pathogen. Then we have this expertise in the lab to molecularly identify all these enzymes involved in the mycoparasitins. But not only that, because Trichoderma also supports the growth of the plants under normal or stress conditions. Then until now, we have seen a little bit about the capacity of Trichoderma to control pathogens, but not only that, Trichoderma supports plant growth through, well, direct growth promoter without any kind of disease. Here on the left, you can see how Trichoderma is basically inducing the formation of lateral root hairs that allows the courgette plants to grow much faster and in better conditions than the plants that are Trichoderma free. All of these has to do with the, well, as mentioned, with the increased lateral root hairs that ends up in the increased nutrient uptake and basically the increased NPK levels that are taken by the plant. This has really implications. Actually, we are proving this not only in the lab, but also in the field. We are formulating Trichoderma with NPK fertilization, and we are capable to have field data proving that if we can reduce up to the 25% of the nitrogen that we need to implement in the fields of wheat, and still the grain yields are the same. I forgot to include that picture here, but just to tell you that we can really reduce the nitrogen fertilization, about more or less a quarter. Rosa is right now more working in the use of trichoderma as an user against abiotic stress tolerance, and also the same here. I have to say that, for example, here in the right side, you can see at seedling states how the use of different trichodermas that correspond to the different numbers is capable to promote and support the growth of wheat seedling under water stress conditions compared to the control. And very importantly, this is also correlated and proven in the field. Then, as kind of a small summary, trichoderma has a direct biocontrol action through the parasitins, antibiosis, enzymatic and secondary metabolic compounds, and also through the direct competition, because it's capable to grow faster than pathogens in the endosphere, in the rhizosphere, and in the soil. And also, very interesting, is capable to produce some bugs, some volatile organic components that is attracting pest enemies of normal pathogens that of pests that are attacking our crops. We are not so much involved at the moment in this fifth point, but the rest we are really covering all the points. And going a little further from the direct control or the control of different pathogens, we have seen that trichoderma is not only capable to direct biocontrol pathogens, also biostimulate the plants, the indirect control, and also what we are working right now a lot is in the studying of the priming stage. That at the end, what we have seen is that there is an inherent response of these priming states that I will explain with a little bit more of details in a bit. And that is the main topic that we are, or one of the main topics that we are having a look at the moment. Then to start to explain this priming and post-transcriptional inheritance, will allow me to go back for a moment to this classical view of the plant defenses that we are aware. We know that we have this plant cell that is attacked for different fungus and pathogens, and then there is the first line of defense where the PRRs that are located in the plasma membrane are recognizing mumps and dumps, and there is a triggering of what we call the PTI or the MTI immune response. Then the case that trichoderma is already colonizing the roots in somehow is fostering this first line of defense, because at the beginning the plants think that trichoderma might be a pathogen, but actually it's not a pathogen. But nevertheless, it's in somehow activating this MTI response. And we know that, well, some of these pathogens are capable to surpass this first line of defense through the secretion of effectors that are usually recognized by intracellular immune receptors like the NLAs, leading to the ETI response. And in this case, also, trichoderma is capable to enhance this ETI response through basically the immune systemic resistant response and the systemic acquired resistant pathways. Here in this review from Wu et al., very quite recent, we try to postulate how the effect or the action of trichoderma can be integrated in the zigzag model for plant defense. And very importantly, introducing this transcriptional and heritable priming memory. Then we know that basically, if we start at the beginning, we see how plants that are untreated, that is the red line, they have an amplitude of defense quite lower compared to the plants that are treated with trichoderma. Then there is a first line of defense that is enhanced in the case of trichoderma treated plants, that is kind of providing a more robust MTI or PTI immune response. If pathogens are capable to surpass this first line of defense through the release of effectors, then later we have this ETI response. But of course, in the case of the trichoderma treated plants, this ETI response is faster and somehow is more pronounced. And what is much more interesting from a molecular point of view is the post-challenge priming defense and the transcriptional memory, because we have seen that the trichoderma plants, the plants that are treated with trichoderma, they are remembering through transcriptional memory this state of defense, then somehow is preparing the plant for a further pathogen attack. And most importantly, it passes this capacity to the offspring through the inherited memory. And then we have here what is called a heritable priming, that in practical use, what means is that if we take the seeds of plants coming for the offspring of plants that are treated with trichoderma, in somehow they are kind of vaccinated against pathogens, at least to a little higher level than the plants that are not treated with trichoderma. And here is where we are trying now to study this at the molecular level, and where we are trying to join forces in the group, because we are focusing now more in the indirect biological control capacity of trichoderma, and we are doing this with biotrophic fungus that are affecting wheat plants. Then there are many of these pathogens. I'm very specialized in rust and power mildew, and right now I'm just presenting you like a little bit of data, how we are studying and what is the effect of trichoderma protecting against these foliar pathogens, because until now most of the research has been focused in pathogens affecting the roots, right, and the necrotrophs. And then, well, we just basically have some wheat plants that are growing under conditions of trichoderma, and later we just simply take some leaves, we make detached leaf segment assays, and under control conditions, we infect with different isolates of power mildew, different races of the pathogen. And what we have seen until now is that some of these trichoderma, they don't have effect controlling with power mildew. Then here, you don't have to go into detail, but basically the control leaves are the ones that are with the blue square, and the plants that were grown under trichoderma conditions, for example, in this case, trichoderma 140, you don't see basically difference. And here I just put a heat map where you see that the color scale that we see in the control section is more or less the same that in the treated plants. Then there is no effect. But we have seen that other trichodermas, for example, the famous Trichoderma 34, has effect in the output between the interaction of wheat and wheat power mildew. Then in the top, I'm showing here one wheat cultivar that has been infected with different isolates of wheat power milieu. And you see that for 4 of them, there is a complete resistance. And for 3 other, no. Then here we have kind of a very specific interaction and effect of Trichoderma 34 controlling the infection, but only against some of the isolates, not towards all. And more importantly, or more puzzling, was to find out that some Trichoderma, the same Trichoderma 34 with another wheat variety that is resistant to many isolates, it creates the opposite effect. It somehow makes more susceptible the wheat plants against the pathogen. Then we have here what I call this bivalent effect, that Trichoderma can induce resistance or can induce virulence. And here in the heat map, I'm just showing how there is this opposite effect where we can go from susceptible plants here that are in white to very resistant in red, in green, or in the other way around. Then we are now having a look at the molecular level to this interaction, because we have here a very high degree of specificity in this interaction that is race specific. And the fact that it's race specific makes me think that NLRs in the plants are involved. And more likely, Trichoderma has these NLRs as a molecular targets, or host targets, that is creating this bivalent effect. Then, well, to conclude the presentation, our group has pioneered in the study of the molecular dialogue between Trichoderma and plants. We are not only working in the lab, but also doing applied science, like, for example, the registration of the first biofungicides in Spain. We have seen that Trichoderma we are studying how Trichoderma is inducing this systemic defense against pathogens, but not only that, we can use Trichoderma to reduce the amount of nitrogen that we have to use in our fields, and this is really important in the umbrella of the current Green Deal. And also, we are seeing how Trichoderma is not only a biocontrol agent, but promoting the growth against or under abiotic conditions, like, for example, drought stress. And this very last thing that I saw, we are studying the molecular basis of the effect and control of Trichoderma against biotrophic fungus in wheat, for which we are searching for candidates for the current Marie Curie postdoc fellowships. And then if you are interested, please contact us. And with this, I would like to thank and to say, well, we have Enrique and Rosa. Rosa is leading the group, and Enrique couldn't attend to this webinar. And if you have any question, I'm happy to take an answer.
Thank you so much, Javi, for your very nice presentation. If there are any questions, yes, let us know in the chat or unmute and just present them.
Naomi, I have a question to Javier.
Yes, go ahead.
Yeah. Thanks, Javier, very much. Very, very interesting, very exciting research you have. I was wondering whether Trichoderma also affects plant mycorrhization or interactions with nodulation. Could you hear me?
With the nodulation, you said?
Yeah, whether Trichoderma also affects in a positive or negative way plant interaction with mycorrhizal fungi,
That's a
or
pity
with the rhizobacteria.
That's a pity that Rosa has to left, because this is really one of the research lines that Rosa is doing now with a PhD student. What I can tell you, because I overheard, I overhear in the lab, is that it has an effect. Then they are doing mycorrhizas with wheat and also combining with Trichoderma. I cannot answer you if it's promoting or is kind of damaging, but I have the feeling that it's promoting, and it's also a strain aspect. It's depending which kind of Trichoderma you are using. Maybe you have a promotion of the nodulation or no, but in theory, yes, it is involved.
Thank you.
I also see a question popping up. Rafa, do you have a question? I'm sorry.
No. I have question, but I will prefer to.
Because I see a question
in the chat. Yeah, you have a question in the chat.
Yeah, Carmen, thank you for your question. I mean, I cannot really tell you. We think it's epigenetics. I mean, we are really doing it now. We are preparing the material and we are doing all the studies, but I cannot really answer you at the moment. I mean, I don't know. But it's happening. At the beginning, when Enrique was mentioning to me, I couldn't believe, but it's really true.
Any other questions?
Yes,
I have another short question to Javier. Are you aware of any Trichoderma effector proteins that could be actually, responsible for the ISR or SAR type responses?
I mean, I cannot tell you. I mean, this is the main goal that we have now in the regional project. There is an effector like I like it to say effector like, because until now, if I'm not wrong, there are not really like true effectors isolated from Trichoderma, at least the canonical effectors that I'm used to see in biotrophic pathogens. But the fact that we have such a specific interaction when I include this with Powell milieu pathosystem and we put the Trichoderma, I only see that when there is an effector involved. Now, if I'm seeing the opposite effect between the wheat and when I keep the same wheat and the same pathogen, the same race of the milieu, but I see the opposite effect, it has to be because there is an effector-like protein or component from Trichoderma, because it's the same Trichoderma, that is having this effect. Then hopefully not in the next webinar, but in 1 year, we can tell you molecularly the idea of the effector.
We're now unmuted.
Javi, if you allow to me, I have several questions actually, but I will do only one. It's not exactly related with effectors, but you consider the peptides that the Trichoderma can produce to allow colonization, or maybe induce this immune response later. I know so the Trichoderma can produce and release some kind of small peptides orthologous to those that plants can do it. So I'm producing. So just a curiosity or an idea,
I mean, that's a possibility, Rafa. The question, I mean, the problem, I'm just going back here. I mean, the question here is that, I mean, these peptides, so far I understood, I don't know if they go inside, but when I have a look to the zigzag model, I mean, the NLRs are specifically recognizing effectors, that this is what we know from, plant immunity. Then I have the tendency to think that it's more effector-like components from Trichoderma. I don't know if there are peptides involved there, but the peptides until now, they have been not described to be recognized by NLS or to act as
I mean, I would say another layer, no, like more in perceiving the membrane level, not intercellular.
Yes,
Yeah, to see if they could be also be involved.
I will not discard this.
Not
for example,
necessarily for. Sorry.
Look, for example, here in this case, you are giving me an idea, because here basically is what is happening is that the same weight against so many different isolates lose the resistance. Then here, maybe there is a peptide that in somehow is, how to call it, is disconnecting one of the PRRs. I don't know, or making it susceptible.
I give you an example. I know, for example, that Trichoderma can produce RALF peptides. They have been known the possibility to produce that.
Really? Okay.
So just as an idea.
Well,
So
I will
it's something.
it.
It will not, let's say, directly related with the effector trigger immunity, but could be also inducing that. So just to, because I mean, I'm working also with raw peptide, and I just for by curiosity, I'm not working exactly with Trichoderma, but I know that they can do it. So maybe we can talk in the future, just to.
Yeah, sure.
So,
We'll be happy.
Okay, I think maybe it's time to move to the next speaker. So many questions for you. I'm sure I have more, but we have limited time. So thank you, Javier, again for your very nice presentation. And now.